Now, something else that we introduced in our recent paper was the concept of consensomes. And what consensome analysis does is it surveys over all of the experiments that map to a specific node family. Using the example here, we're going to use insulin receptor. So we'll select consensomes, we'll select receptors, catalytic receptors, and the family of insulin receptor. And we click submit, and we see our consensome report. And using the example here of this insulin receptor is to rank the genes by strength of their regulatory relationship with nodes in that family. So what you're looking at here on the left are the list of genes that are consistently regulated in insulin receptor interrogated experiments. You see a discovery rate. Now this is the frequency with which that gene is differentially regulated in those specific experiments. You also have a geometric mean fold change, giving you an idea of the magnitude up or down. And you also have a consensome p-value, gives you a sense of significance that the result you see is not due to chance alone. And then we rank these genes into percentiles. So, a representation that isn't available on the site right now, but one that I think makes this idea easier to understand is plotting these points in a scatter plot, where on the x-axis you have the discovery rate, which again is essentially the frequency over all of the experiments in which that specific messenger RNA was significantly differentially expressed. And on the y-axis here you have the rank of that gene in the consensome itself. So typically up here at the top, you've got genes that are highly responsive to the insulin receptor, less so at the lower discovery rates here. And at the bottom, you have those genes that are very weakly or are unresponsive to insulin receptor signal. Now, just as a technical note here, you're going to notice this interesting two-tailed distribution that we see in many of the consensome scatter plots, where genes of the same discovery rate actually have different consensome p-values and therefore different rankings. The reason for this is that genes in the upper tail were assayed in larger numbers of experiments than those in the lower tail. Therefore, they're assigned a lower p-value due to the increased sample size. So when you look at consensomes, this is one caveat to be aware of. Now, so by ranking genes based on their responsiveness to a specific node, consensomes offer us a unique environment where the user can actually use SPP to formulate new hypotheses that they wouldn't have been able to using only the information in research literature. So what we've actually found, having looked now at consensomes from many different nodes, and again, using the human insulin receptor here as an example, is that the most highly ranked genes, in addition to those containing those that are known targets of the insulin receptor, we also see many genes that have no evidence of being involved in insulin receptor signaling. And so information like this can help researchers hypothesize novel mechanisms of action and signaling systems that have not been explored in the research literature. So to exemplify that concept, we're going to look at the relationship between insulin and autophagy. So what is autophagy? So autophagy is the process by which cells, in response to depletion of their extracellular nutrient sources, attempt to maintain energy levels through metabolism of nutrients within the cell itself. And this process is actually well known to be regulated by insulin, which is, of course, the primary signal of the fed state. Now, insulin regulation of autophagy has been very well documented in the research literature at the protein level through regulation of AMPK. Now, interestingly, and this is something that has not been explored in the research literature, key factors that are involved in the regulation of autophagy are actually highly ranked in the insulin receptor family consensome, indicating that they are transcriptionally very responsive to signaling through insulin receptor family. And these include, for example, ULK1 here, ATG14, also known as ATG14L. And in fact, 33 of the autophagy pathway genes, of the 33 autophagy pathway genes in the human genome, nearly 50% of those are in the 90th percentile of the human insulin receptor consensome. So this leads us to the hypothesis that in addition to regulation of autophagy at the protein level, signaling through insulin receptors supplements that level of regulation by modulating the expression of messenger RNAs and coding factors in the autophagy pathway. All right. So now here, if you're looking at the regulation report for the most, well, here first let's filter on ATG14, one of those autophagy genes that was highly implicated in insulin receptor signaling. Now, if we click on that gene, it takes us to a single gene transcriptomic report for the most highly ranked factors. And remember, it maintains our filters that we initially picked for our consensome. So what we're looking at is a single gene search of AGT14 for catalytic receptors, insulin receptor family. But what's very clear is that insulin signaling exerts a profound negative effect on that gene's expression. Multiple different insulin receptor family member agonists all repress expression of ATG14. But what is equally, if not more striking, is that over half of those data points, the ones you see here in gray, those are far below the twofold cutoff that is typically widely used by investigators when they evaluate results of an expression array or RNA-seq experiment. So if an investigator was looking at this experiment right here, for example, in isolation, he'd not be very convinced. The fold change is 1.3, even though that p-value is significant, still it's not a very big fold change. However, if you take the sum of all the data points which you can visualize in SPP, now a very clear and consistent pattern emerges, and that is the autophagy gene ATG14 is downregulated by insulin receptor signaling.